Morning. I wanted to make a few announcements for the class. The announcements are written on the board to my right, to your left. The first announcement is that several of you asked me what the role of the textbook is. Will I take exam questions from the textbook? And again, I want to emphasize to you the textbook is meant as a reference. It's got a lot of good pictures in it, and it's helped you to clarify concepts which we may only touch upon here, like meiosis, for example, if you don't remember it. So no exam questions will be taken from the textbook for my portion of the midterm. It's possible that the same subject matter is discussed here and in the textbook, in which case then you could say an exam question is taken from the textbook. The second announcement is that I had to cancel the office hours yesterday. I'm very sorry. It was a last-minute emergency I had to attend to. And so I'm going to have office hours tomorrow to make up for those from 9:30 to 10:30 for those of you who were unable to make, because I wasn't there, the office hours. And finally, there will be sample questions from previous midterms posted on the website for the class either today or tomorrow. We have plenty of time before the first midterm, but at least this will give you some idea of the way you ought to be thinking. And of course, I've asked you questions here in lectures which parallel the kinds of questions you will be getting on the midterm. Today what I want to do is to introduce you to the first organ system that we'll be encountering in plants. Plants do not have very many organs, and the one we're going to talk about today for our first organ is the root. I don't know. Can I turn up my mic? Let's take a look here. I don't think so. Oh, maybe they've turned it up. I think he's turned it up. So there, okay, you're all set. It might be too loud now. No, it's not too loud? Okay, great. So the first organ system that we want to consider today is the root system. And the root system is the part of the plant that most of you never think much about. It's buried, you think, in the ground, and sort of it fits the old saying, out of sight, out of mind. Now, the general functions for the root system, which we're going to talk about today, some in more detail than others, are the first function that we're going to discuss is support of the plant, or anchorage, sometimes called. The second function of the root that we'll touch upon is that it is a site for storage, usually of starch, sometimes sugars. And we as humans have exploited this capacity of roots to store starch and sugars in the form of various crops. So, for example, here is a beet. Beet is an enlarged root system in which there is sugar or starch stored. Carrots are an enlarged root system which you like to have sweet, where there's sugar, perhaps some starch as well. Here is a root system that may not be familiar to all of you in the class. This is cassava. This produces a very, very fine starch, which in the tropics is a major component of the diet of many of the people who live in the tropics, cassava. And finally, a root system which is not consumed, but which has a sort of a cultural value, is this root system from a plant called ginseng, mostly used in East Asian cultures. The shape of the root system often dictates how it's used in various ceremonies. For those of you who've purchased ginseng before, you know this root system can be quite expensive to buy in the stores. So roots function, as far as we're concerned, many of you encounter roots every day in the form of foods, and that's because they are a storage organ. The third association or function of roots that we want to talk about is that they are the site of absorption for water and nutrients. And this is an extremely interesting function, and we're going to be spending most of our time talking about how this absorption of water and nutrients occurs from the soil. And then finally, although not last, which we might have time to touch upon, is that roots establish symbiotic associations with many of the soil microorganisms. And this serves to benefit the root system and the whole plant in general, and it also presumably benefits the microorganisms who are in a symbiotic relationship. Now, when the seed is first germinating in the ground and the seed coat is cracked open, the very first structure to originate or to penetrate the seed coat is the root. And we call this first root, which comes out of the seed, the primary root. And because we're going to mention primary so many times, I'll abbreviate it with a 1 with a little 0 up at the top. Now, in a eudicot, the primary root grows straight down and it dominates the root system. In monocots, the primary root forms. You'll get a primary root just as before. But the primary root is short-lived. And dies and is replaced by roots from the stem. So this replacement of the roots from the stem, where the primary root dies in the eudicot, results in a shallow root system. Is that for us? No. Okay. A shallow root system. And this shallow root system, because it lacks a primary root, is very fibrous in nature. Fibrous in nature. It's almost net-like as far as its appearance. I'm going to show you some pictures of these in a moment. Now, because of this fibrous and shallow nature of the monocot root system, for those of you who come from areas where there are forest fires or fires on hills where the grasses are burned, what you will find is that following the fire before the winter rains, you will see helicopters or airplanes flying over the burned land and spreading or depositing monocot seeds. And the idea behind this is that the seeds will germinate with the first rains, and because they are shallow and fibrous, that they will knit the soil together and thereby prevent erosion. Now, if you were to go out and look at root systems, because they are buried under the ground, they're not easy to characterize, not easy to see. And for this reason, the first slides I'm going to show you are root systems which come from the tropics. In the tropics, many of the root systems of plants are actually aerial. They are above the ground, and they can show us some of the variety. So first we're going to look at some slides which show us pictures of root systems from tropical plants, and then we'll look at some slides from root systems growing under the ground. So the first slide is of a plant which is known as screw pine or pandanus. This is a monocot, and its primary root has died, and it has been replaced by roots which are growing from the stem of the plant, tropical plant. Here's another tropical plant. These structures here look like they're trunks of trees, but in fact, they are root systems growing down from these long arching branches, and they act as support for the branches. Here's a very interesting root system which is found in trees that are large, that grow in areas that are very windy. This is called a buttress root, and it allows the tree to remain upright in very windy conditions. For those of you who come from Florida or from areas where there are mangrove swamps, you know the swamps have the tide coming in and out several times during the day. And here is an example of a mangrove tree growing in a swamp in which the tide is out. And notice how the root systems elevate the tree above what would presumably be the level of the water when the tide comes in. Now, sometimes the tide comes in and the root system is different. It doesn't elevate the tree. And here is an example of another type of mangrove in which the tree is not elevated above the ground, but in fact what the root system does is produce upward growing roots. And these upward growing roots go up above the level of the water, and it is believed they act as passageways for air to go down to the root system to allow respiration to continue, even though the root system is technically flooded. Now here's a root system that some of you can probably see outside, although may not be aware of it. This is ivy, a climbing ivy, and ivy is able to climb up because its root system secretes a very strong glue, which, as far as I know, has not been analyzed, so that if you try and strip the plant off of the building that it's growing on, the root systems usually remain attached. Here is a root system of a palm tree, and in this particular root system, this is a monocot. These are roots forming on the stem. The root system has been turned into sharply pointed thorns, and it is believed that these downward pointing thorns prevent animals from climbing up the tree and eating the fruits. This is the root system of a plant which grows in a desert environment, a very hot environment, and the plant, which you'll actually see when you go to the botanical garden, is able to pull itself down into the ground so that it is not exposed to the heat or to predation by these roots which are known as contractile roots. They actually contract and pull the plant into the ground. For a person who studies roots, this plant represents probably the ultimate plant, because this is an orchid plant in which most of the body of the plant is a root system. This is a plant which grows in the light, and the root system is in light, and it is green because that's where photosynthesis goes on. Here are the plants that we just saw before. We have got carrots, cassava, and beets. Again, these are ones in which we as humans make a lot of use of the root system. Root systems can be very damaging. These are pictures inside of a sewer. Most of the damage that roots do, aside from breaking foundations, is by growing into water pipes or sewers and blocking them up. Here is ginseng, this plant here, where the shape of the root system is believed to confer some type of indication of what it can be used for ceremonially. Now, let's take a look at the root system. I guess I'm going to continue. Let's take a look at the root system of a germinating seedling. This is a radish seedling in which the primary root is the first root that comes out. Here you can see the root hairs, which we discussed before, and we'll spend some more time talking about them shortly. Here is a corn plant. A corn is a monocot, and its primary root has been replaced by these roots. Many of you have seen these structures before, growing from the stem. If we look at a corn plant which has been excavated, you can see the root system, and notice that there's no one root which is predominant over the other. There are some shallow roots and some deep roots, but the primary root has died and been replaced. Unlike this alfalfa plant, where the primary root is very evident, very obvious. Notice that we have a lot of so-called branch roots or lateral roots, and we're going to spend a few minutes talking about how they originate. Now, as I said, it is very difficult for one to excavate a root system, but a good example turns out to be work done in England. This is, I think, an apple tree, and in this particular case, you can see the root system of the tree and the shoot system, and notice that the amount of biomass below the ground and above ground in this particular case is almost equal. We're going to return to this idea in a moment. And finally, I just want to show you this picture taken of a plant and the root system in part of this plant from Southern California. This is the plant over here. Here is a person, and this individual has excavated one of the roots from this plant to show you the area in which, how far this root can go from the plant and how much it can exploit the environment. And one might guess for a plant like this one that there's much more biomass of the root system below ground than above ground because the plant needs the water. Okay, so these are the root systems, and I want to mention now a few things. We can go ahead and put the screen up, done. I'll mention a few more things about the root systems. From the pictures that you saw, you could see that the one from the apple, that the biomass making up the root system and the shoot system was about equal. This ratio changes as the plant ages, so that if you look at a plant which has just germinated, you have much higher biomass in the root system because it's important for the plant to establish a strong root system before it makes a shoot system. For those of you who are gardeners or who have parents who are gardeners or grandparents, if they are good gardeners, when they transplant the plant, that is when they dig it up from one place and move it someplace else, what you're doing is you're severing the root system. You're leaving roots behind in the soil. and you're told what you should do is also cut part of the shoot system. That is, to restore the balance between the root system and the shoot system. So this balance is very tightly controlled and is important in determining the success of a plant in its environment. Now what I'd like to do, having given you this brief introduction to the variety of shapes and sizes of roots, is to talk about root development. And root development is a fascinating topic, and we're going to give you some reasons why it's a very currently a hot fashion in plant biology. And for our discussion of root development, I want to begin with this model of a root system. And I want to point out to you on this model of a root 2 zones that we're going to spend some time on. This yellow zone, which is located from your perspective at the tip of the root, and this brown zone. Now, if we give names to these 2 zones, we could call this the root proper, and the structure on the end, so let's make a picture of this. We have the structure which is yellowish on the end. This is the root proper, or the root body. And the structure on the end is called the root cap. These are 2 separate regions of the root. They comprise the root that we're going to spend some time talking about. Now, one can also take a root and slice it down the middle. And in slicing it down the middle, you'll be able to see a section of the root cap and of the main body of the root. And that section is known as a longitudinal section, or sometimes abbreviated just as LS. And on the other side of this root, which you will also have in the lab, you can see a longitudinal section through the root proper, the brown part over here, and through the root cap, the yellow part over here. One other region I want to add on here as far as a landmark that we'll be referring back to is that in this region, just behind the cap, we're going to put it on here, and I'm going to move this down here so it's not in your way. In this region, just behind the cap over here, the other landmark which we're going to go back to is the landmark called the zone of cell division. Or you could call this the root meristem. Now what I'd like to do is to spend a few minutes talking about each of these 2 parts, the root cap and the root body. And I'd like to begin with the root cap, which is a fascinating structure. Charles Darwin and his son Francis spent a lot of time studying the root cap, and in fact, they called the root cap, as you will see in a moment, they called it the brain of the plant. So let's look first at the root cap and discuss their functions. And this really is only going to be the tip of the iceberg with regard to the functions. Now, as you can see from this model, the root cap is at the very tip of the root. So the very obvious and first function we want to just note with regard to the cap is that it protects the root meristem as the root grows through the soil. So the root cap functions in protection of the meristem. You can imagine there are lots of rocks in the soil, a lot of interference with regard to the root growth, and the meristem can be damaged. The cap essentially takes the brunt of that damage and protects the meristem. How does it protect the meristem? Well, in part, it secretes or produces, let's say, a lubricant, again, which remains unanalyzed, a lubricant which actually eases the root's way through the soil. This lubricant is referred to scientifically as a slime. And finally, the function I want to put down now, but which is not meant to be a comprehensive list of functions of the root cap, is that the root cap is the site of perception of gravity. And I'd like to now discuss with you this very intriguing mechanism. And for this discussion, what I'd like to do is we'll draw the root but we're going to really focus on the root cap. And the first thing I want to point out to you into understanding this mechanism of how gravity is perceived in the root cap is to note that there are groups of cells in the root cap which contain membrane-bound starch. These organelles which contain membrane-bound starch are called amyloplasts. And these cells which contain the amyloplasts are called statocytes. So let's talk about how these amyloplasts are involved with the gravity protection, the gravity signal. Starch is heavy. So if we could magnify, which we will do, one of these cells here from this root, one of these statocytes here from the root, we would find that the amyloplasts are located on the lowermost side of the cell. Because they're heavy. They're located on the lowermost side of the cell. So here is the root, just to give you, this is the orientation of the root, and the statocyte would be like so in the root. Now, let's take this root, and instead of having it being oriented in this vertical plane, let's change its orientation to gravity. And for this discussion, what I'm going to do is to call the sides of the root, this lowermost side we're going to call B, and we're going to call this vertical side in its original position A. And now what I'm going to do is change the orientation of the root so it is now like so. We've turned it 90 degrees. So in this particular case, if we looked at this cell here, what we would find is its orientation has been changed so that now A is on the lower surface and B is now on a vertical surface. When the orientation is changed from the root's vertical position, the starch grains begin to fall. The amyloplasts fall to the new lower surface. And we think this displacement or falling of the amyloplasts to the new lower surface is the beginning of the signal for perceiving gravity. This tells the root your orientation to gravity has been changed. Now, what would you expect would happen to the root if you take off the root cap? Well, if you remove the root cap, no ability to detect gravity. Or the gravity vector. Now, what might you guess would be the situation which has actually been done where they have sent roots up into space, either on the space station or on the shuttle, and you have the roots sitting in the space station? Which direction would you think they would grow? Well, the answer is that the roots grow in all directions in the space station. There is a small amount of gravity called microgravity, but not enough for the root systems to detect. So they grow in random directions, and often this direction is determined by which way the astronauts happen to have moved the roots to impart a small force. So in microgravity, or in this case we'll call it zero gravity, or in the absence of the cap, the starch grains are not able to move. They're not able to detect where the new gravity vector is because there isn't any. And in this particular case, when you're in microgravity, the vector can be any direction. The root is unable to respond to gravity. So this is an important aspect of the root cap's function, is that it actually determines where in the soil, and it isn't always straight down, as you can recall from some of those roots. Some are growing horizontally. But the signal of gravity is processed to allow the root system to be able to either grow straight down or grow at an angle. Now what I'd like to do is to spend a few minutes and not look so much at the root cap, but to spend time looking at the body of the root. So let's begin with a longitudinal section of the root again. We put the root cap down here. Here is the zone which we previous of cell division, or the meristem. And then what I want to point out to you on this root, and I'll move it down here, and this is why roots are intensively studied by developmental biologists and why they are very fashionable subjects for plant molecular biologists. If we look in other regions of the root, what we will find is a little further back from the meristem, we find elongation of cells. And a little further back from that, we find that the cells are differentiating into the cell types that you know of. So here in this one axis, and this is why roots are of so much interest to developmental biologists, in this one axis, one has these three developmental activities occurring in a single root. You've got the cells being formed, you've got the cells that were formed elongating, and subsequently you have the cells differentiating. This is a very important system that are used by developmental biologists. Now what I'd like to do is to begin to take us to the function of the root, namely the function of the root as a site of absorption of water and nutrients. And in order for us to appreciate this really fascinating mechanism by which this works, we have to look a little bit more at the cross-sectional anatomy of the root. Because if something is going to be absorbed, it's coming from the outside, and it's going to have to move in to the root and be absorbed there. So let's talk about the simple anatomy of a typical eudicot root, and all the cell types you will know of except for two new ones. So what we're going to do, instead of making a longitudinal section of this root, we're going to make a cross-section of the root. So let's begin with a cross-section, and I'll just abbreviate cross-section, of a typical eudicot root. We'll begin, easy part, making a circle. And let's put some landmarks on, many of which you know already. In the very center of the root is the vascular tissue. The very center of the root, and we'll talk more about that later on. That would be the xylem and the phloem. On the outside of the root, again, the tissue you know about, the epidermis. You've already seen this before. Most of the root is comprised, up until the epidermis and up until near the vascular tissue, most of the root is comprised with a thick layer or thick region of cells which is known as the cortex. Known as the cortex. And there are only two other cell types which we have to put on. Both of them are new to you. The first cell type is located around the vascular tissue, and it is called the pericycle. And the reason this is an important layer is this is because this is where branch roots form. We're going to go back to that. I'll show you some pictures of that in a moment. The only other layer we have to put on is this layer, which is immediately outside the pericycle. This layer immediately outside the pericycle, which we're going to be spending a lot of time talking about. And this layer immediately outside is known as the endodermis. Endodermis. So this is a typical cross-sectional anatomy of the root with the features that we have to know about in order to understand this critical function of absorption of water and nutrients. Now what I'd like to do is to just do a high-power view of this region over here and add a few more points that we're going to have in our minds when we talk about how a root absorbs water and nutrients. So for this picture, I'm just going to put the vascular tissue in the center, like so. Here we will have, for our discussion here, the endodermis. And now what I'd like to do is to make a series of cortex cells. Until we get ultimately to the epidermis. So this is the epidermis. And this is the cortex. Now there are a few features that I want to put into this very high-powered view of the cross-section of the root. What you need to know, and this is an important part for understanding the function of the root, is that all of these cells are alive. If they're alive, that means they have a membrane. Thinking back to when we talked about living versus dead cells before. So I'm going to use this yellow chalk, which may not show up on the video, to line each of these living cells with a membrane. And we have the endodermal cells too. The only other feature we need to put onto this view is that going from one endodermal cell to the other are very small submicroscopic pores that some of you have already discussed with me that we can't see with our eye. In fact, you can't see with a light microscope. So there are pores, and these pores run between cells. And these pores are called plasmodesmata. They're pores which run between living cells from one cell to the other. They're essentially tunnels connecting one cell to the other, but they're very tiny. Now let's do an experiment where we begin to think about the uptake of water and nutrients into the root system and talk about how this works. The first thing I want to say to you is the root is growing in this soil environment. The soil consists of a very complex mixtures of minerals, of toxins, of other substances which may or may not be good for the root. The root has to decide, it has to make a decision, and this is a decision which requires energy, whether or not it's going to take something up from the soil. For example, the roots like to take in calcium because calcium is a very large component of the plant body. But just like us, roots want to exclude sodium. So how is it that this living system, which is buried in the soil and surrounded by potentially good as well as potentially toxic nutrients, how is it that the root is able to discriminate and to decide which of these two elements get into the shoot system? In order for us to appreciate this, I want to do an experiment for you. And in this experiment, what we're going to do is to have a beaker. And in this beaker, we're going to dissolve two different species of ions. And we'll just call them A and B. And now we're going to put a plant into the beaker. Here's a plant in the beaker. And allow it to begin to absorb or to take up A and B. And we're going to wait now for some time. Let some time pass. And we're going to ask, where are A and B at the end of some particular time? Where did A and B go? And for this experiment, what we're going to say is for this particular plant, after we allow some time to go by, if we look for A, we would find that A makes it all the way to the top. But if we look for B, what we find is that B is only in the root system. only in the root system. Why? And what does this tell us about the mechanism of selectivity and absorption of the particular species by the root system? In order for us to appreciate this mechanism, I'm going to now take our typical root and add a few more cells onto it so that we can see where A and B are in this particular root system. So again, we'll put our vascular tissue in the center. And now I'm just going to take a series of cells, cortex cells, which I'll draw here for you. And here we've got the epidermis again. And we're just going to put another layer of cortex cells on here. And then we have maybe one over here, and then an epidermal cell. So if we look to see where A and B are in this particular system, what we find is that A is in all the cells and in the vascular tissue, which accounts for it to get up to the top. If we look to see where B is, what we find is that B can be found in these holes that are between cells known as intercellular spaces. And they can be found in cell walls. Now, how can it be found in the cell wall? The cell wall, although it may seem solid to you, is really a matrix with lots of pores and holes. So what we're saying is that you can find B in this root system either in the intercellular spaces or in the cell walls. So if we look to see where B is, it would be potentially in all these places, maybe in the cell walls. And the innermost part that we find B is that B stops moving just outside the endodermis. Stops moving just outside the endodermis. So now let's look at where A and B are with more resolution. If we look at this particular drawing here, and we put A on, let's say we see A inside this cell here. How did A get inside of this cell? In order for A to get inside this cell, it had to cross a membrane. Had to cross a membrane. So in order for it to get inside, it had to cross a membrane. Once it is inside the cell, A has access to all the cells inside by going through the plasmodesmata. There are also plasmodesmata here, eventually ending up in the center. So once it enters a membrane or crosses a membrane, it can then travel through the plasmodesmata inside the cells. The pathway which A follows includes crossing a membrane, and then traveling through the interior of the cell. We call this pathway, which involves crossing the membrane and traveling through the interior of the cell, we call this the symplastic pathway. You've got to cross the membrane in order to enter the symplastic pathway. How about B now? What do we see happens to B? If we look carefully about where B is in this particular diagram, as we have mentioned in the diagram over there, B only remains outside the membrane. So B is outside the membrane. It never crosses a membrane. So remaining outside the membrane is one of the characteristics in this particular case of the species B. So that eventually B accumulates at the endodermis. Can't go any further because it's outside the membrane. Now, why is that? Well, first of all, like the pathway which involves the movement through the membrane and the inside of the cell, which we said was the symplastic pathway, the pathway which is outside the membrane and includes only intracellular spaces and cell walls, we call this the apoplastic pathway. Never crossing a membrane. So here we have B in the root, but for some reason, B is stopped in its movement into the root, making it all the way to the endodermis, and then it stops. It can no longer go any further into the root. Why is that? This is where the plant then exercises some decision-making with regard to what it takes in. If it wants to ship an ion or a species of ion up to the top of the plant, that ion has to cross the membrane. If it doesn't cross a membrane, it cannot get any further in the root system, in theory, than the endodermis. Now, of course, there are some poisons and toxins, and there are also some ions that the plant really doesn't want that get in by mistake. But in general, this system functions very, very effectively to take in the ions the plant wants and to exclude those that it doesn't want. Now let's think about and examine what it is about the endodermis that stops the movement of the ions into the root. And this, conceptually, is probably the most difficult part of the lecture. What we can say is that in the wall of the endodermis is embedded a material which is water impervious. This is the same material that makes cork float. It's called suberin. Now, this is the part that's hard for you to understand, and I want to make sure that we are all here together. Here is the endodermis located in this particular region here. And what I'm saying to you is that if this is the thickness of the cell wall of the plant, this is the thickness of the cell wall, embedded in the cell wall is this material called suberin, which is water impermeable, or water impervious. So that if an ion is in the apoplastic pathway, it can move into the cell wall here because there are lots of big pores for it to go through. But as soon as it gets to the layer of suberin, which is also on the inside of the cell, it extends all the way through this middle region. As soon as it gets to the layer of suberin, it stops. It can no longer go any further. So it is this layer of suberin which prevents further movement of ions into the root unless that ion has crossed the membrane. If we pretend like this balloon is the membrane, then, of the endodermal cell, and we stick it in this diagram over here, you can see that if you are in the symplast already, the endodermal cell has its plasmodesmata connected to the plasmodesmata of the cortex cells, and as an ion, you can go all the way through the root, through the balloon, and end up in the vascular tissue. But if you are in the apoplastic pathway, you are not have ever crossed a membrane, and so you're going to be stopped right at this point over here. Very important point conceptually. This is what this suberin layer does. It allows the plant to discriminate and to decide only by letting something cross a membrane will it take it into the inside of the root. This is the important point. Now, this material, which is known as suberin, is also given a name. It is called the Casparian strip. This green layer that surrounds the endodermal cells, the Casparian strip. And it functions in preventing movement from where you are in the class, in the apoplast, to where I am in the center part of the vascular tissue. It's very important. Ions cannot squeeze through. They can't get in unless the plant lets them cross a membrane. Now, an ion can cross a membrane any place. It could go all the way in the apoplast up until the endodermis and then cross the membrane here. And once it gets into the symplast, then it has access to the vascular tissue. So it doesn't have to enter the symplast at the outside. Crossing any membrane, any time, allows you to move into the symplast. Finally, I have two sort of cartoons here. I'm going to put the screen down while I'm showing these to you. Can you put the screen down? If you could imagine each of these two cells as endodermal cells, and the little blue dots on there is plasmodesmata, and you are sitting outside in the cortex and I'm in the vascular tissue, in order for you to get in, and these orange strips represent the suberin or Casparian strips, in order for you to get to where I am, you have to be inside the symplast of these particular endodermal cells. You can't be in the cell wall, because if you're in the cell wall, you haven't crossed a membrane. And keep in mind that this endodermis extends all the way up and all the way around the root. It is a cylinder, a seal, which prevents ions from sneaking through in case the plant doesn't want them. So this is a very effective, very efficient system, but it depends upon the plant exerting energy at the membrane to either take an ion in or to keep an ion, to exclude it. And it's not fail-safe, as many of you probably can guess. Some things do get across. Let's look now at some pictures of this particular situation. And the first ones I want to remind you of are the root cap that we talked about. Here we have the root cap with the slime that you can see. And if you take a root and you put it on its side, the root bends down. And think of this as being time. And if you take a root and you remove the root cap and you let the root grow, it essentially doesn't perceive gravity. Here are some pictures of the amyloplasts in the statocyte cells. And when the root is reoriented, the statocyte cells, as you can see in the next slide, change their orientation with respect to gravity, and the starch grains fall to the new lower surface. Here are some electron micrographs showing you the statocytes, and these other organelles here are believed part of the response. Now here's a cross-section of the root showing the cortex and this large region here that the ions have to pass through, whether in the apoplast or the symplast. And in this particular view, you can see the endodermis here and the vascular tissue in the center. So in this particular case, the ions move to this point here and then are stopped in their movement. Finally, before we look at the apoplast and symplast, I want to show you the branch roots which come from the pericycle. These are formed in precise arrangements, and it means the root has to grow out from the pericycle and through the cortex to get out. And finally now, let's look at the apoplast and symplast. What I want you to see on this slide, particularly, are the intercellular spaces. Through these intercellular spaces and the blue in the cell wall, the B pathway, the ions can move all the way to this point and then are stopped. Only when they are inside the cell, in the symplast, can they go further. These are the plasmodesmata. This is taken with an electron microscope, very tiny. You can't visualize them with your eye. And we now have plasmodesmata running between these cells and allowing the symplasts of individual cells to be connected. So an ion is absorbed by the root hair and then moves through. I think this is somehow supposed to be moving. And then, we're not going to stay that long. And then I want to show you this 3D view of this. This is an important view because it's very hard to get three-dimensional views of this. This is the outside of the root. This is the Casparian strip on the endodermis. And only if you are inside the symplast, so pretend like you're an ion right at this point here, you would be able to look up to your right and to your left and below you and see the Casparian strip. It's not a curtain. It's only embedded in the cell wall. And so although you were surrounded by the Casparian strip, you are not prevented in your movement from the outside of the endodermis to the vascular tissue in the center. And here is another picture just trying to diagrammatically show that. Here is the cortex. Only if you're inside the symplast can you move into the vascular tissue. And not to belabor the point, this just shows you the two pathways. And this pathway is important. I wanted you to just notice this slide here, because in this particular case, this arrow over here has moved all the way by the apoplast, all the way to the endodermis, and has stopped. The ion can still get into the root, but it has to cross into the symplast of some cell before it can then enter the vascular tissue. So make sure you understand this very important mechanism. This is the way plants operate. This is the way they discriminate between what they take in and what they exclude.